Hey everyone, this is Emmett Hightower, or Sam Hightower actually. This is my cell project. I made a prokaryotic cell out of other than elementary school play-doh. Because I have no baking skills or not really creative like that. Made a simple bacteria cell, or we're going to make a simple bacteria cell, and this species is Chryseobacterium greenlandensis. Okay, this is a bacteria that I read about in a Time magazine article a couple months ago. It's found in Greenland. It's been around for more than 120,000 years in the ice in Greenland and survived in extreme cold temperatures and really changed how everyone thought life can live in the planet Earth. Okay. You're going to start off with the cell wall. It's going to be the black Play-Doh here. And it's made of peptidoglycan, and it gives the shape and protection, and it contains the pili, but we'll get to those later. The pretty cool part of the bacteria. And then we also have the cell membrane, which will be the orange part of our Play-Doh here, that goes inside here. I'm just kind of like put that in there, and so you kind of have like a dual layer. Alright, now the cell membrane is very similar to eukaryotic. It's still got a phospholipid bilayer, and it's still semi-permeable, selectively semi-permeable. And it helps with energy conservation because bacteria are so inefficient. But instead, you know how eukaryotic cells have a lot of proteins, they're going to have a lot of fatty acids inside of them with transport and unsaturated fats and saturated fats and whatnot. And now you're going to have the pili, which are these little protein tubes that have adhesive tips that help the bacteria connect to other bacteria and to the host cells and whatnot, and pretty much cover all over the bacteria. There'll be these little green things, long and cylindrical, and just cover everywhere. And for the sake of time, I'm not going to sit here and put these everywhere, but you get the general idea. Okay. I was going to fall off on me. But, you know.
It's so funny.
Yeah. Okay. And that's what those are. Now they would cover the entire bacterial cell. And notice how this only has a cell wall and a cell membrane. There's also such thing as a capsule, which would even cover over the cell wall and the pili, and it's pretty much just going to be a carbohydrate and stiff, rigid extra layer that will help protect even more, so other bacteria won't eat this bacteria. But this bacteria doesn't have it, so we don't need to include it in this presentation. Next up would be the cytoplasm if you're going to keep on going along, but for the sake of being able to see a model of everything, the cytoplasm, we won't really actually have a particular Play-Doh color or anything in here, but you'll just imagine everything inside of here is going to be in cytoplasm, and that's just pretty much going to be a gel-like matrix that is also in eukaryotic cells, and it I don't know why I pronounced that funny. And yeah, it protects and keeps all the organelles inside of it. Alright, next up is the DNA, or the nucleotides or whatever that's going to go inside. And they're pretty much the genetic information. This is how it's arranged in a prokaryotic cell or bacterial cell. There's no nucleus at all. It just free floating in the cytoplasm, and it just kind of sits there. I don't know if you can see. I don't know if I've showed it too well. There's the pili, two different walls, black, orange, and then the red DNA. Okay. And then last up is the ribosomes, which I have these little bitty teal-looking bits of play-doh that I'm going to stick just on the sides, but they actually float around and they're freestanding. And what they do is translate the nucleic acid into amino acid to help make proteins. Now, you will notice how in eukaryotic cells they will be, everything's falling off, attached to the endoplasmic reticulum as well as float around, but these are completely freestanding and do whatever they want in the cell. Now, bacteria reproduction. Well, you've got ribosomes there, little teal things. Okay. Bacteria reproduce through binary fission. This falls out once again. It's not going to stay very well. Okay, and this would definitely be in the first stage where the chromatin are still being reproduced, and that hasn't really split up yet, and it's not going to like pinch apart yet. It's still getting copied and reproduced, and still in one cell, and hasn't really started forming into two like different bodies, and there's going to like slowly pull apart. And the downfall of binary fission is if you're going to get two completely identical copies of the same bacteria, they're both going to be susceptible to the same diseases and antibodies and stuff. So that's how we're able to find cures of bacterial diseases and infections. And thanks for watching my video, and now you know what a prokaryotic bacterial cell looks like from 120,000 years ago. And it's a very simple form of life and very small and different than the standard eukaryotic cell. Have a good day.